Welcome to our podcast on the respiratory system. As we begin looking at this system development, it's going to be one of our first ones now that we've moved out of the pharyngeal apparatus. We're going to see that we are going to form the bulk of the respiratory system, certainly our trachea and our lungs, as a branching off of our foregut endoderm. So the lining of our respiratory tract is going to be endodermally derived, and you may see a lot of different cells that are going to form from this endoderm. And you can see how we're going to branch off of our foregut. That's what we see in orange. And as we're going to expand this tube and branch a number of times into all the branching that we see in our adult respiratory system. Now we'll get into a little bit of molecular biology as we get to the end, some more inductive forces. But first, let's see where we're going to be doing this. Now, again, this is going to be in the foregut region of our developing embryo. So, our larynx, trachea, bronchi, and lungs, they're going to begin to form during that magical 4th week of development. Now, again, we're going to be looking at, here we have a sagittal cut through our embryo. And then if we make a cut, again, like the cut we were seeing when we developed our oral cavity in this, here we have our pharyngeal arches, here we have our tongue buds, our lingual swelling. This hypobranchial is what it's going to be termed as in many places. You're going to see in this new edition of the book, they call it the hypopharyngeal, which is actually a better terminology. This is going to be a part of what's going to form the pharyngeal part of the tongue, but this is also going to assist in forming a part of the epiglottis. Now we're going to see a little groove that's going to form back here in this most posterior part of our endoderm in our pharyngeal region. And this groove just behind this hypopharyngeal eminence, that is going to be the opening that's going to lead to the respiratory tract. Now in comparative anatomy, when you looked in the amphibian, that's referred to as the glottis. And we can still refer to that. But here in developmental terminology, so we're going to call this the laryngotracheal groove. And this is going to be really the beginnings of where we're going to enter into our pharyngeal apparatus. Now let's watch a video that's going to, again, give us orientation and a perspective of where exactly this is happening in the embryo. As we begin forming our respiratory system, as we look at the floor of the pharynx, we're going to begin to form a blind-ended pocket, a diverticulum. It's going to invaginate downward, and it's going to extend to form the trachea, the bronchi, the lungs, all those passageways. And so the endoderm is going to give rise to the epithelium and glands of the larynx. Now, when we say glands, we're talking about secretory cells, and we'll talk about those just a little bit. But as you can see, larynx, trachea, bronchi, pulmonary epithelium, that's going to come from the endoderm. But the connective tissue, which is going to include cartilage, smooth muscle, this is going to come from the surrounding splanchnic mesoderm that's going to be induced to form these structures by the endoderm in the correct place. So here we're going to see at the very lower part, the laryngotracheal diverticulum. This is going to be connected to the laryngotracheal tube, the laryngotracheal groove that we saw earlier. And this is how we're going to access from the foregut, we're going to access our respiratory tract. So you can call this the laryngotracheal diverticulum. Now, as we begin, we're going to see that the end of our respiratory diverticulum is going to continue to expand. It's going to grow, and it's going to turn into what is referred to as the respiratory bud. Now, some people call these lung buds. Your textbook will as well. I like to refer to this one as the respiratory bud, because what's going to happen is this is going to be the trachea, but as we get lower and lower and extend it further and further, this is going to branch into our two separate lung buds. That's how I sort of think of them. So once again, we can see in this illustration we have the yellow endoderm and the red surrounding that is going to be representative of our splanchnic mesoderm that's going to be induced. Now, we can see as we go down, we've extended that diverticulum. We're going to change its name. We're going to call it the laryngotracheal tube after we extend it. And at the very bottom, you're going to take that lung bud, or the respiratory bud, and you're going to branch this into the bronchial buds. Now, these are going to give rise to your right and left bronchus, these passageways that we have. And so as we extend this on downward, we're going to expand the laryngotracheal groove, and it's going to open up, and they refer to that as the primordial laryngeal inlet. And again, think of this as the glottis almost as much as anything else. So here we can see our laryngotracheal groove, and now we're going to expand that. We're going to have our primordial laryngeal inlet. We can see the laryngotracheal tube. They're calling this the trachea at this point. And then our bronchial buds, some people refer to these as the actual lung buds, since we now have the right and left portions. But for me, bronchial bud is going to be the most characteristic, the most descriptive of these two. Whereas the first one, I like to use respiratory bud for that. Now, in this area of the primordial laryngeal inlet, what's going to happen if we can take a cross section? Here we have our foregut. Here we have, if you look down at the bottom of the screen, our primordial laryngeal tube, laryngotracheal tube. In this area, we're going to have these folds begin to push in. And these are going to be our tracheoesophageal folds. These are going to fuse together, and these are going to help to partition off the foregut from the laryngotracheal tube, or our laryngotracheal, or our trachea per se. And this is going to adjust the height and the size of the opening so that we have that in the appropriate place. Now, these tracheoesophageal folds, they're going to continue to grow more and more along the midline until they fuse together, and they're going to form the tracheoesophageal septum. Now, this is really going to be coming from the inferior portion of the foregut, sort of moving up, pushing, if you will, just the perspective, moving that primordial laryngeal inlet higher and higher and redefining its shape. But as we form this tracheoesophageal septum, that's where we are going to effectively separate the laryngotracheal tube from our foregut, which in this case is going to be the precursor to the oropharynx and to the esophagus. Now, once again, just as a reminder, the epithelium, the lining of the larynx, the lining of our trachea, this is all going to be endodermally derived. Now, in the larynx, we have a lot of cartilage that's going to be present. And in this case, our laryngeal cartilage is going to be from our 4th and 6th pharyngeal arches, and to a large degree, this cartilage is going to be from our neural crest cells. Now, we're back to looking at the floor of the pharynx in that cut open view. Here we can see our laryngotracheal tube. Here we can see our hypobranchial eminence. And what's going to happen on the lateral sides of our laryngotracheal tube, we're going to have these areas of high mitotic activity. They're going to enlarge. And the mesenchyme is going to proliferate. And these are going to be the arytenoid swellings. That's what these are called. So as we form these arytenoid swellings, we can see our hypobranchial eminence is beginning to extend further and further back. We're going to convert our laryngotracheal groove from just a slit. We're going to convert that into a T-shaped opening. And the upper part, this more cranial part, if you will, this is going to be converted into our epiglottis. So once we form our T-shaped opening here, part of our primordial laryngeal inlet and get that T-shaped, that's really when we're really going to start to refer to this as the glottis. And the upper part here, this cranial part, is going to be our epiglottis. Here a little bit further in development, again, we can see the arytenoid swellings are pushing this higher and higher up, really creating that flat T-shaped as the epiglottis and the hyperbranchial eminence, well, hypopharyngeal eminence, is growing backwards, pushing down on this part to flatten out our T-shape and form the epiglottis. Now, we're going to put this together into an animation of the formation of our primordial laryngeal inlet and the formation of the epiglottis. So as we've extended our laryngotracheal tube and we form our bronchial buds, we're going to see that in the region of the larynx, we've formed our primordial glottis, we've got our epiglottis, but the lumen is going to be temporarily closed. We're going to have cells completely occluding this area, and it's not going to open again until 10 weeks or so of development. Now, as we open this, as we have apoptosis occurring to canalize this passageway, we're going to see that we're also going to form what are referred to as laryngeal ventricles. These are recesses. And so you're going to have portions of the wall of the larynx being eroded, again, by apoptosis. And what's going to be left are these folds of mucous membranes. We're going to have the vocal folds, which are your vocal cords, but we're also going to have the vestibular folds. Now, these are going to be just superior to the vocal cords. They're going to be much more structurally significant, and they're going to be a protective shelf that's over the vocal cords. So if anything happens to get into your airway, it's not going to immediately damage the more flexible, the more fragile, that's the word I'm struggling to find, the more fragile vocal cords. Now, we saw the epiglottis. That's going to form at the caudal end of that hyperbranchial eminence. And as the name implies, this is above the glottis. It's going to protect the glottis. Now, the epiglottis, recall through anatomy, that as you drink fluids or you eat solid food, what we're going to see is that the epiglottis is going to cover over your glottis and almost provide like a ramp so that this material can slide back to the more dorsal esophagus. Now, the larynx and the epiglottis, they're not going to reach their final adult position until about 3 years of age. And that's important because newborns, as you watch them, they can suckle a breast, so they can drink milk and breathe through their nose at the same time, primarily because of the structure of the pharynx and the laryngeal region. So as we begin to move down, now we get into our trachea development. Again, the endoderm. This is going to be our epithelium and glands of the trachea. We're going to see that the splanchnic mesoderm, again, is going to comprise and form our connective tissue, the cartilage and the smooth muscle. And it's at this point, as we begin to form that tracheoesophageal septum that's going to separate the esophagus from the trachea, we're going to see if we have an inappropriate segregation of the septum, we're going to form a tracheoesophageal fistula, an inappropriate connection between the respiratory tract and our digestive tract. Now, as we look at how some of these can actually form, we have, we've got our respiratory diverticulum. There we can see, again, the bronchial buds, lung buds. I like to refer to these separately. Here we have our septum. that is moving up, but we can have situations where instead of moving this opening up higher, having the esophagus continue on down, we have an inappropriate connection. Here we see in the superior part, we've got a normal esophagus. We've got a normal, basically primordial glottis coming into our respiratory tract. But look, we got a couple of things going on. Here we have the fistula, the inappropriate connection between respiratory and digestive. But in this case, we've got an esophageal atresia. It's completely closed off. So the only way that this newborn is going to be able to feed is for material to pass into the respiratory tract, and hopefully some go here. This is not a good situation, not compatible with life, because too much fluid and food is going to be aspirated down into the respiratory system. This has to be surgically corrected. So here we have just another illustration showing tracheoesophageal fistula, that inappropriate connection. Here we have a before picture, then after surgery. And again, that has to be done fairly quickly before any damage can be done, because babies are going to need to eat right away. Breathing is not going to be a problem. It's when they feed, and babies eat all the time. Now, as we get to the end, we get to our bronchial buds. Again, some folks call these lung buds, but let's just stick with the first one off of the foregut being the respiratory diverticulum, or the respiratory bud, and these two lower ones being either lung buds or bronchial buds. Okay, that will just keep it simple. I know the book might describe it in these other terms, but let's do it that way. So we have our bronchial buds that are going to form. And again, these are going to grow into this area where we have our pericardio-peritoneal canals. Now, this is where we're going to form our pleural cavities for the adults. So they're moving into this area. And again, if we have a posterior lateral defect in our diaphragm and we allow abdominal contents to come up, there's not going to be space for these to expand. We already saw that with our pulmonary hypoplasia. So our bronchial buds, these are going to give rise to the primary bronchus, the right and left side. And again, the epithelium on the inside is endodermal. The cartilage on the outside, the connective tissue, the smooth muscle, that is going to be from our splanchnic mesoderm. Now, as we look at the lung, we're always going to have 3 lobes on the right side, 2 lobes on the left. So we have more lung tissue, which means the right bronchus is always going to be larger in diameter. And if you have small kids that are coming into the ER, they've aspirated something, well, the first place you're going to look is the right bronchus, because this is the place where foreign material is most likely to enter and get stuck, more so than the left side, because the left side has a smaller opening diameter. Now, the primary bronchi, much like we talked about in phys, the primary bronchi, they're going to branch and they're going to become secondary bronchi. Some refer to these as stem. The right, we're going to have superior, right superior, and we're going to have a right, 2 of those. We're going to have right inferior, and these are going to go to the middle and the inferior lobes. And on the left side, since we have the less space, we're going to have 2 superior and 2 inferior. So that's our secondary bronchi. Now we're going to get into the tertiary. It's another level of branching, and we're going to have 17 orders of branching. We're not going to name them all. But by this 7th week, we're going to have tertiary segmental bronchi, and they're going to be 10 on the right side, and there are going to be 9 of these in the left side. Now, if we look at that segmental bronchus, that tertiary that we just talked about, plus the surrounding mesoderm and the lung tissue that forms all of these elements, then we can refer to that as a bronchopulmonary segment. These are going to be isolated one from the other. So if one bronchopulmonary segment gets damaged, then you are not going to affect any of the other areas, even on the same side of the lung, maybe on the right side. So bronchopulmonary segments are isolated by connective tissue septa, so if that part of the lung gets damaged, you're not going to lose function in the other segments of the lung. And again, as we mentioned, those 17 orders of branches, and that's going to be by week 24. This just illustrates a little bit, looking at cross-sectional views through these. We can see our trachea, primary bronchus, stem bronchi, which are secondary, and then you see sort of the prospective look of where the lobes of the lungs have formed at this point. I don't think we see our bronchopulmonary segments yet in this particular illustration, but they're going to form as we expand and further branch our airway in the lung. So now we have a video showing the development of the trachea and this branching. And then next, we're going to look at some of the molecular aspects of why and how these cells know when and where to branch. We're now going to focus on the cellular mechanisms that allows this to happen. We have induction, we have chemical signals, and we have differential cell division, and that's principally what's going to be happening here. So as we look in lower organisms, and in this case, if we're looking in Drosophila, they have these tracheal-like tubes. We're going to see that we have mesodermal cells, and that's going to be illustrated here in purple. And these mesodermal cells are going to secrete an inductive agent that is called branchless. Now, this branchless is an FGF-like protein. It is a growth factor. FGF stands for fibroblast growth factor. FGF is going to signal the underlying endoderm to undergo more rapid cell division. As the more rapid cell division occurs, we're going to extend out. Okay, here we have a branch tube. It's growing closer and closer to the inductive center. We're following a concentration gradient until we pass a threshold, at which point in the threshold, the cells that are at the the cells that are at the most concentrated point are going to secrete a negative feedback mechanism that shuts down branching along the sides and is going to increase the mitotic activity at this point. At which point, when you get increased mitotic activity, you've inhibited on the side. Now you're going to get a secondary set of branches. So this was seen in Drosophila. Now, as we look in higher organisms, in mouse, for instance, what we're going to see is that we do find FGF10 that is produced by the mesoderm. In this case, we've got mesoderm again in purple. Here's our FGF10. But what's going to happen as we begin to branch, and as our endoderm gets closer and closer, that gradient gets higher and higher, we're going to reach a point that the endodermal cells are going to begin to secrete this green material, and this is sonic hedgehog. SHH is the abbreviation. Sonic hedgehog is going to provide a negative feedback to the mesodermal cells and cause them to stop making FGF10 in the central point, But the lateral cells still making FGF10 will cause the increased activity on the sides, and there is a branch point that occurs. So that's how branching occurs. Now, as we get back to our developing lungs, we're going to see that we've got our trachea, we've got our primary, we've got our secondary, we'll eventually get our tertiary, as we see here, in our endodermally lined portion, and then our red mesoderm on the outside. And what we're going to see on the outside of the lung itself, this outermost layer, that is going to become our visceral pleura, the actual mesothelial covering that occurs on the outside. On the body wall, we're going to have a mesothelium, a very thin mesodermally derived layer. And this is going to be our parietal pleura. This is going to provide this low friction surface. This is our intrapleural space. And this is where we're going to see that pressure difference that causes the visceral pleura to continually be pressed against the parietal pleura. It's not going to adhere, it's not going to stick, but because of those pressure differences on the inside of the lungs versus the outside in the interpleural space, it's going to be pressed against it at all times. So it's going to create a potential space. Now, as we look at lung maturation, we've got our airways in place. By the 24th week, remember, we've got 17 orders of branches. So now as we look at our stages of development, we're going to be looking at things like the extent to which alveoli have formed. That's where we're going to get our respiratory membrane and gas exchange occurring. We're going to be looking at capillaries, the different cell types that are present as we define these different stages. So we've got pseudoglandular, canalicular, terminal sac, and the last is the alveolar period. So from 5 to 17 weeks, this is our pseudoglandular period. We basically have everything in place for lung function, except we don't have the membranes in place, the very thin portion, the association with capillaries, so no external respiration is going to occur. And prior to 17 weeks, if there's a premature birth, there is zero chance of survival because of zero gas exchange that's occurring. Now, as you notice, there's a little bit of overlap. So that one was 5 to 17. Here's 16 to 25, and this is just sort of numbers we're throwing out. There's going to be a range depending upon the different developing fetus. So the canalicular period, this is where we're going to see a lot of our capillaries begin to form. So this highly vascularized nature of the capillaries. Also, the branching nature, we're going to reach that 17th level of branching, and we're going to begin to see these terminal bronchioles. And remember, terminal means that this is the last part of the conducting pathway before we move into the segments that can actually lead to gas exchange. So here, we're going to be seeing those respiratory bronchioles and the alveolar ducts. We may even, towards this 24th, 25th week, begin to see some of these alveolar sacs, terminal sacs, that might have some of these beginning, the primordial alveoli. We might see some of these begin to form. So at this point, 25 weeks and earlier, we can't say 0% chance of survival, but the chances are very poor because we simply don't have the final adult alveolar structure in place. So here we see pseudoglandular period. Really, we don't have any thin membrane. We do have some blood vessels. They're not very closely opposed to our capillaries. That's where the gas exchange is going to occur. Here in the canalicular period, you can see many more capillaries. You can see they're becoming more closely associated with the pathways. But it's out here in these terminal sacs where we need to see that close association of capillaries with a very thin membrane. And we just don't see a lot of that yet. So the survival at this point, we say, is very poor. So the cells that line our airways, we've got our epithelium, and at this point when we're looking at our bronchioles, even our terminal bronchioles, they're going to be columnar or cuboidal in shape, kind of square. It's by the time we get into our respiratory bronchioles and then our alveoli, this is where the cells become very flattened and thin, and that's really what we're looking to happen as we go from these multiple layered passageways down into our single layered passageways of tall cells, shorter cells. We've got to get into the really, really flat cells before gas exchange can occur. Now, in some of these early pictures, you can see these are fairly thick-walled airways that are passing here. This is a bronchus. This is our respiratory bronchial, and we really don't see any of our little bubbles, the little pockets of alveoli or even terminal sacs here that we can really say gas exchange would occur in. So that's why prognosis is very, very bad for premature babies born before 25 weeks. Now, the terminal sac period, 24 weeks to birth. This is where we have the capillaries very close to the alveoli, the very flattened epithelial cells, our type 1 alveolar cells. Squamous is a term that means flattened like a fried egg. So these flat cells are very close to the flattened cells of our capillaries and these endothelial cells, and that is our blood-air barrier. And it's thin enough that gas exchange is going to occur. We also have type 2 alveolar cells, or these giant alveolar cells, that are producing surfactant. And we'll talk a lot more about surfactant. But this phospholipid mixture, a detergent, if you will, this is going to break the surface tension so that we can open up these little tiny sacs called alveoli so that we can have the correct amount of surface area for gas exchange to be effective. Now, once again, you see the overlap. Okay, we get to this maturation period, and the terminal sac period, that is going to be the 24 weeks to birth. And so here we've got, sort of two different settings as far as prognosis goes. 24 to 26, very poor survival still, but 26 to 28, it's better, but not great. You want to definitely get past that 28 weeks. And really what you're looking to see, and the reason we have these ranges, you need adequate pulmonary vasculature. You've got to have sufficient numbers of capillaries, and you have to have a significant amount of surfactant being produced. If that's produced at 24, your survival is going to be better. If it's not produced at 28, your survival is going to be low. So there's still a lot of variability as far as prognosis with premature babies in this range, depending upon those two really important variables. Now, our last stage is the alveolar period. This is late fetal period through birth, childhood. Basically, we are refining our alveolar capillary membrane. This is our blood-air barrier, the type 1 pneumocytes, and our capillary endothelial cells. You definitely have it thin enough. Our alveoli are going to mature after birth because, remember, before birth, they're simply bathed in amniotic fluid. So when we look at the factors that are critical for adequate lung development, certainly we already know, based on what we studied with the diaphragm, you've got to have thoracic space. If you have any impedance from the abdominal contents, you're going to have pulmonary hypoplasia. Fetal breathing movements, bringing that amniotic fluid in and out. An adequate volume of amniotic fluid to do that. You're exercising the muscles that are going to be involved in ventilation. That's critical. Now, in the alveolar period, we've got a real great chance of survival here, but our problem at birth, our lungs are half filled with fluid. And the way we clear those is through the mouth and the nose during birth. As you're squeezed through the birth canal and the head's delivered first, your thoracic cavity is compressed, and so that's going to be basically expressed through the mouth and the nose. And you'll see a lot of neonatal nurses with an aspirator sucking out the mouth and the nose even when just the head has been delivered. We're going to have the absorption of fluid through pulmonary capillaries and our lymphatic drainage. That's going to remove the rest, but it's going to take a little bit of time before we can remove all of that liquid. So here we see the development through our periods. The pseudoglandular, the canalicular, the terminal sac, they refer to as saccular here, and then the alveolar. We see the overlap. We see the production of surfactant. And then below, we can see where we're really going to be producing in the end of our branching pathways. And once again, we're going to get the alveolar sacs, and this is where our survival is particularly improved. So now let's watch a video that talks about the development and the different periods of development of our lungs. And now, as we've mentioned earlier, surfactant, and this is just what I want to cover just as we finish up this unit. This lipid-like complex, what's going to happen is that the surface tension of water inside these very small alveoli would tend to collapse the alveoli down. The air pressure needed to keep them open would not be sufficient against the surface tensional forces of water, and they would all collapse. So essentially what this hydrophobic mix does is these phospholipids, the hydrophilic part associates with the water, but the hydrophilic tails keep the water molecules far enough apart that the surface tension is virtually eliminated. Now, surfactant, this basically comes for surface active agent. That's how the term was combined. And basically, when you look at these lipids, the dipalmityl phosphatidylcholine, this is the principal phospholipid that we have present. And as you look at this molecule, you can see the charged head on the right. You can see this long hydrophobic chain, this hydrocarbon chain. And in this transmission electron micrograph, we can see the large giant alveolar cell. This is type 2 alveolar cell. These are the lamellar bodies within that cell. And these are the surfactant molecules that have been released into this solution. It's going to coat the alveoli. This is the alveoli that's coating. And the significance of this can really be seen in the law of Laplace. And we're going to get to that in a second. But when we look at pulmonary surfactant, we can see that, number one, we're going to prevent the alveoli from collapsing, but we're going to increase pulmonary compliance. Now, this term is the volume change that's going to occur per unit pressure across the lung. And as we eliminate the surface tensional forces, we're going to really have a low pressure requirement compared to the volume that we're going to change. Here's a for instance. If you look in the lower left, this is an alveoli with surfactant, and the pressure is sufficient. You basically need zero pressure to keep it open. Without surfactant, it's going to be completely collapsed. It's going to be impossible to keep that open. Now, the law of Laplace basically summarizes how this happened. In the law of Laplace, this is the air pressure needed to keep an alveoli open. And the pressure needed to keep an alveoli open is basically going to be proportional to the surface tensional forces and inversely proportional to the radius. As the radius decreases, this pressure is going to increase. But with surfactant, this factor, T, the surface tensional forces, is zero. So regardless of the radius, the pressure needed to keep this alveoli open is going to also be zero.